Good morning, everyone. Today, myself, Dr. Richard Singh, and today I'm gonna here for you guys to share part 4 on the topic cell, the basic unit of life. And today we are gonna finish that chapter for sure. So the remaining topics was in this topic was like, so first we need to write the heading cell, the basic unit of life. As you can see, I've written the heading. Now, the topics in today's lecture we are going to cover are as follows. First is going to be mitochondria. Yes. The second one is plastic. Third one is like ribosomes. Ribosome. Fourth one is cytoskeleton. Cytoskeleton. Fifth one is cilia and flagella. And the last one is nucleus. So here guys, here we are, and today we are going to complete these 6 topics in today's lecture, and after that this chapter, the cell, the basic unit of life, is going to be completed for today and for sure. So let's come to the topic. So the first topic is mitochondria. Let's just write a subheading, that is mitochondria. Mitochondria. So when I say mitochondria, what you people understand by mitochondria? Yes, you have heard about it. It is actually the powerhouse of the cell. Powerhouse of the cell. So you know this thing very well, that the mitochondria is the powerhouse of the cell. So let's just draw a schematic diagram of a mitochondria in order to understand the basic headings and the basic terminology which comes under this topic. So here is the mitochondria. This is the longitudinal section, and that is longitudinal section, and it's like this wave-like appearance inside the mitochondria, like this, like this. So here we go. So there's thing inside this is like this. This is circular DNA which is possessed by the mitochondria, and there's also one cell organelle other than mitochondria which also possesses the circular DNA, and that is plastic for sure, and that is the second topic. So here we are. Let's draw some ribosomes. Ribosome. These dots are ribosomes, and again, so let's just draw, let's just write the headings. So this outer is membrane, is outer membrane, because it is outside the mitochondria, so it is the outer membrane. Okay, and this thing, this wavy appearance is also a membrane, but which lies beneath the outer membrane, so that is why it is called the inner membrane. Inner membrane. Okay. Now this black dots are like, these are ribosomes. This red thing is circular DNA. Let me just write it down with the red pen. This is circular DNA of mitochondria for sure. So again, this space is called intermembranous space. Intermembranous space. Okay, and this thing is called matrix. So okay, so what is matrix? So matrix is just actually the space between the inner membrane and the outer membrane is also known as lumen or lumen. So I can write in lumen. And the space which is inside the inner membrane is called as matrix, in which various chemical reactions occur. Actually, I have already explained this to you. The mitochondria is the powerhouse of the cell, and so basic work of mitochondria is to generate power for cell. So that is why it will be generated by some chemical reactions. So all the chemical reactions occur inside the matrix of the mitochondria. So let's just talk about the size of the mitochondria. First point is the powerhouse of the cell, and the second point is it is about 0.2 to 1 micrometer in diameter and 1 to 2 micrometer in length. But that is so true, because mitochondria can be of variable sizes and variable shapes. But actually, it is the schematic diagram, it is the basic mitochondrial structure. But the shape and the size of mitochondria can be changed. If it is present in some cell, it could be of different shape, and if it is present in some other cell, it could be of different shape. So it doesn't matter. But the thing is, mitochondria in every cell is also show variability in the number also. Some cells have more mitochondria or some cells have lesser mitochondria. So number is also variable. So as you can see, number is also variable. And this red thing is single circular DNA. Single circular DNA, that means mitochondria also have its own DNA. So, and mitochondria divide by the means of fission. Fission is just division, simple division. Mitochondria just split from between two halves and just divide into two, and one mitochondria give rise to another mitochondria. So that is how the fission works. So here is the next point. It divides by the help of fission. Is that clear to you? Sure. So the thing is, that is the mitochondria, that is the powerhouse of the cell, and it is the diameter is 0.2 to 1 micrometer and 1 to 2 micrometer in length, and it divides by the help of fission. Okay, mitochondria can be of variable sizes, variable numbers, depending upon the type of cell which is in which mitochondria is present, and it also possesses a circular DNA, the DNA of its own. And if I talk about the ribosome is the protein synthesizing organelle. It is actually the membrane-less organelle. The special type of ribosome which is possessed by the mitochondria is 70S type. So here I have written. So that's the basic structure of mitochondria. So let's come to another topic. Okay. Well, actually, the thing is, mitochondria helps in generation of energy in the form of ATP. So here I write another point. Where should I write? Here's the space. It generates energy in form of ATP. ATP stands for adenosine triphosphate. Okay. So how does this thing works? Many people ask me, how does mitochondria produce energy? Well, actually, it helps in respiration. Respiration is a chemical process. Respiration is related to breathing, but actually breathing is not particularly just respiration. Respiration is chemical process, and the breathing is the physical process. So after we inhale oxygen, we exhale carbon dioxide, we inhale oxygen. So oxygen comes into play in generation of energy. So if I just remove these points, I will make you understand the thing, how the mitochondria do the thing. That is respiration. So actually, the thing is, there's one cycle called as glycolysis in which glycolysis, 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 as the name suggests, glyco means sugar and lysis means splitting. Okay, so that is the basic understanding you need to have right now, because actually glycolysis is the topic of some other chapter. So we are gonna understand that thing in the next chapter, that is respiration in plant. So glycolysis, the basic knowledge you need to have right now is just glycolysis. Glyco means sugar and the lysis means splitting. Means sugar is getting split. So sugar, the basic sugar we talk about is C6H12O6. That is glucose. Is that everything clear to you? That is glucose, that is the basic carbohydrate. So it requires oxygen and it gets converted into 6CO2 plus 6H2O plus energy. So this is the energy we need. And this is the basic oxidation and oxidation reaction of glucose. So glucose requires 6 molecules of O2 and say produces 6 CO2 plus 6 H2O and plus energy. Now the thing is, there is a reaction called glycolysis which occurs in the cytoplasm. It occurs in cytoplasm. Okay, and glycolysis convert glucose into pyruvic acid. So that is the whole purpose of glycolysis, which occur in cytoplasm. So it converts glucose to pyruvic acid. So again this pyruvic acid comes into play, and this get translocated inside the mitochondria, and after that aerobic respiration occurs. So aerobic respiration is the cyclic process which occurs in the mitochondria, where pyruvic acid is converted and ATP production occurs. So ATP is produced by mitochondria, but it is a halfway process, because the pyruvic acid is generated in cytoplasm and the rest of the process is gonna occur in mitochondria. So that is the whole thing. That is the basic idea you need to have right now. So I need to rub this thing. I hope you understand what is the work of mitochondria. So this glycolysis thing we are going to read in some other chapter, that is respiration in plant. And after that, so mitochondria is complete. Now the next topic is our plastid. Okay, so what is plastid? So plastid is also a double membranous structure like the mitochondria we already just understood. So let me just make a basic diagram of plastid. Plastid is of three types. Plastid basically of three types. And what I am making is the structure of chloroplast. It is like this, and there's also a DNA, and this is ribosomal structure, and again this is again this pile of coin-like structure like this. A lot of these kind of structure are present in chloroplast. So the plastid, actually the basic thing is plastid is a kind of a structure which is only present in plant and some euglenoids and in some algae. So the plastid are of three types. Here is the space I am writing types, three types. They are named as chloroplast, The next one is chromoplast, chromoplast, and again leucoplast. Okay, so these three types of plastids are present in every photosynthetic or maybe in green plants, some algae and some euglenoids. So here we go. So I'm going to explain each and every one of those plastids. So first we need to understand, first we need to just label the diagram which I have drawn. So this is outer membrane. This is inner membrane. And this is again the DNA. This whole pile of coin-like structure is called granum. Singularly we can say grana. And this simple structure, this single structure is known as thylakoid. This bridge which connects granum with granum is stroma lamini. Okay, and again this dot-like structure is ribosome. Ribosome, a special type of ribosome, is present in plastid of chloroplast. Actually, it is the diagram of chloroplast. Chloroplast. Okay. So, special type of ribosome is present in chloroplast is of 70S type, but actually, whenever we see same, actually 70S type is same as the ribosome of mitochondria. But when we talk about eukaryotic cell, you can see it is in which 80S ribosomes are present. So here we go. It is the schematic diagram of chloroplast, and plastid are of three types, and it is present in each and every plant, each and every green plant, some algae and some euglenoid. Okay, and structure and number can be just like mitochondria. The number can be varied according to the organism. If it is unicellular, maybe one chloroplast is present, or if it is a green leaf or a tree, maybe 20 to 40 chloroplasts could be present. Okay, so plastids are of three types. First one is chloroplast, which I have already drawn on the as a diagram on the board. Second one is chromoplast, and third one is the leucoplast. So according to their name, their functions are also different. Chloroplast, it helps in. So where should I write? I think I should just remove this thing, and I must write here. So chloroplast. So the function of chloroplast is it helps in trapping light energy, trapping light energy and produce food for plants. Okay, so the thing is, chloroplast help in trapping light energy. It uses light energy to produce food for the cell. So that is the basic function of chloroplast. And another one is chromoplast. As the name suggests, it is responsible for color because chromoplast, it is responsible for providing color. Actually, in chloroplast, inside the chloroplast, a pigment called chlorophyll is present. So here I am writing with red color, chlorophyll pigment is present. Is that clear to you? So that chlorophyll pigment is responsible for trapping the light energy and convert biochemical energy they are converting into food. So again the chromoplast, chromo, chromoplast, it is responsible for providing color to the plant or the part of plant. So because it provides colors, it has pigments like carotenoids and xanthophyll. Carotenoids like carotene and xanthophylls which are responsible for providing color like yellow, orange and red. So these kind of colors which is provided by the carotenoid and the xanthophyll pigment which are present in the chromoplast of plastic. The third and the last one is the leucoplast. I hope you understand the diagram. I need to rub it in order to explain you the leucoplast. So what leucoplast help in? Leucoplast. So basically it is a colorless plastic. It is colorless plastic. It does not have any color. So it could be of three types. Basically it helps in storage function. So it helps in storage function. So basically the thing is the third plastic, that is leucoplast, it is basically colorless and it helps in storage function. And it could be of three types depending on the type of substrate or organic compound they are storing. So let me explain. So three types of leucoplast. So the names are like this. The first one is amyloplast. That stores carbohydrate. Third one is oleoplast. That stores oils and fats. The third one is aleuroplast. That stores protein. So as you can see, there are three types of leucoplast present in the cell. First one is amyloplast, that stores carbohydrate, and the oleoplast stores fats and oil, and the aleuroplast stores protein. So three kind of leucoplasts are present in the cell, and this was the basic structure of chloroplast, which actually unfortunately I have just cleared. So that's all about plastids. Again, the next topic of our is ribosome. First I need to rub this. So here we go again. Our next topic is ribosome. So as you all know, ribosome is membrane-less organelle. It is also known as organelle within organelle. Here actually there is a one PYQ neat question which is frequently asked. Which among the following is organelle within the organelle? So that is the ribosome. So it is also known as organelle within organelle. So it is a very important statement. So you need to just remember this thing because this is frequently asked in your paper. Actually, ribosome are also of two types. So ribosome are of two types. Two types of ribosome. Ribosome. Namely, the first one is 70S and the second one is 80S. As you can see, the S stands here is the Swedberg coefficient. Coefficient. So the S stands for coefficient. Actually, it is the unit that gives us the measure of the density and the size of. Actually, it is a sedimentation coefficient. Actually, but it is named after the Swedberg coefficient. So it is the sedimentation coefficient. In bracket, I need to write it is the sedimentation coefficient. That explains the sedimentation coefficient and it gives the measure of size and density of ribosome. Okay, so is that clear to you? So this S is Swedberg unit and it is also known as sedimentation coefficient. It gives a measure of size and density of ribosome. Okay, so let me run this thing again. So as you can see, 80S ribosome is made up of two units as well as the 70 unit ribosome is also made up of two subunits. One subunit is bigger than the other unit. So the bigger unit is like, so 70S is made up of like 50S plus 30S. So these two units make like this. Ribosomal schematic diagram is here. So as you can see, the bigger one is the 50S subunit and the smaller one is 30S subunit. So it makes 70S. Whereas when we come to the 80S comprises of the 60S plus 40S. That makes ribosome like this. So this is 40S and this is 60S. So what is the another difference is the 80S, the ribosome are all present in eukaryotes, whereas the 70S are present in plastids, mitochondria and other prokaryotic structures. So this is the basic difference between these two ribosomes. So that is all written in your NCERT. So let's come to the next topic, which is the cytoskeleton. Cytoskeleton is very simple and very easy to understand. So here we come. Cytoskeleton. Okay, so every cell has its own skeleton. Skeleton provides support, sustainability, and allows growth and flexibility also. So in your NCERT, it is explained that it is made up of microtubules, microfilaments, and intermediate fibers. So the thing is, let me explain you with an example. So have you seen a construction work nearby where the huge magnificent buildings are constructed by a contractor? So you can see the basic structure of a unit. The basic building is composed of these rod, pillar or beam like structure, and after that the concrete is filled between these spaces. Have you seen this kind of construction? Yes, I'm sure that you have seen such kind of construction. Basically, the building is made up of beams and pillar. Okay. this. And after that, these empty spaces are filled with concrete and bricks. So if we say that concrete and bricks are the cemental material, so these beams and pillar are microtubules or microfilament and intermediate fibers. So if we say that if it is a cell, it is maintaining its structure like this, and this is the nucleus, and it is maintaining its structure like this, and it need to maintain its shape also. So it must have rigidity in cell wall and cell membrane. So this support, this rigidity is provided by the cytoskeleton, and the cytoskeleton is made up of microtubule, microfilament and intermediate fibers. So that is all written in your NCERT, and that's the basic thing, and nothing more need to understand in this. Next topic is our cilia and flagella. Cilia and flagella. Cilia and flagella are like hair-like structure you have already seen in a bacteria or in some like structure. Basically, the cilia is also for movement and flagella is also for movement. And here the longer tail of bacteria is, this is flagella. And this hair-like structure is cilia. Both these are meant for locomotion. Like bacteria, if he wants to go somewhere, then he must have one flagella or can have cilia. Cilia are longer, so I must write like this: cilia, flagella. Okay, they are short. First difference is they are short. They are long. They are long. All right. They both are for locomotion and movement, and this is the basic difference, and that's all you need to understand. If we cut flagella, if this is the basic structure of flagella, this is flagella, a long filament-like structure. Then this outer covering is made up of plasma membrane. So all I am trying to say is the outer membrane of flagella is also made up of plasma membrane, and plasma membrane is also covering the whole cell. So these two things are made up of plasma membrane. So this is the essentially thing. So the filament like structure that is called as flagella is made up of plasma membrane. Okay, so here we come. If we just take the cross section area of flagella, so cross section of flagella, we see this structure, like this is the cross section of flagella. We see like this kind of structure. This. So we see in your NCERT, this diagram is very clear cut. So we see microtubules are present in flagella, running parallel to flagella. That means I must write. So microtubules present in flagella which runs parallel to longitudinal axis of flagella. So here's the thing, these are microtubules arrangement in the flagella, which is actually in the form of 9 plus 2. We say 9 plus 2. Why 9 plus 2? Because in the outer covering you can see there are uh, doublets. We can say doubles or doublets. So one microtubule is present in the form of doublets, and these doublets are present in in order like 9, and inside this circumferential arrangement, after that it comes, here we see 2. So this particular arrangement is called as 9 plus 2 arrangement. So here I need to write arrangement of microtubules is 9 plus 2. That is also an important question which is frequently asked in NEET. So here I am writing it important. So you need to understand these microtubules are arranged in doublets, and these doublets are present circumferentially in flagella, and they are in number of 9, and inside this arrangement there are 2 microtubules present, which are 2. So this particular kind of arrangement is called as 9 plus 2 arrangement, as you can see. So this is the cross section area of flagella, and flagella is covered by plasma membrane, and cilia and flagella, they are 2 different things, but actually the function of those 2 are same. So here we come to our next topic, that is nucleus, which is the last one, and after that this chapter is going to get completed. If you have any question regarding this, then you can ask me anytime. So here I am writing is nucleus. So nucleus is a very important organelle, and it is present in each and every cell, and it is responsible. So let me write it helps in running. It helps in running cell function. That means it is responsible for viability of cell, or we can say if the cell is not having nucleus, then we can say it could be a dead cell. Okay, so nucleus. So let me make diagram. If this is a cell and this central thing is nucleus, and it is covered by nuclear envelope. Okay, so let me label it first. This is nuclear membrane. This pore is known as nucleopore. It is filled with nucleoplasm, just like cytoplasm. Nucleoplasm. This structure, like a concentrated structure which is present inside the nucleus, is known as nucleolus. If singular, if plural, it is plural. It is called as nucleoli. Okay, and if singular, it is called as nucleolus. If plural, it is called as nucleoli. And the nucleus is covered by nuclear envelope. Envelope means it has 2-3 membranes. First is the inner membrane, and the second one is the outer membrane. Both inner and outer membrane gets fused and make nucleopore. So as you can see, nucleopore is seen because the outer membrane and the inner membrane are getting fused over here, and the nucleopore and makes nucleopore. So the function of nucleopore is that it helps in transfer of RNA for when we talk, if we talk about transcription in which mRNA is comes out from the nucleus into the cytoplasm. After that, translation process happens. So it helps in passing. It makes pass RNA from nucleus. Nucleolus is the concentrated genetic material. Genetic material is present in cell in nucleus in the form of chromatin. As I can make you understand like this, chromatins are like these fibers. These is genetic material called as chromatin. Chromatin. So chromatin. It is the genetic material. Genetic material. As we all know that chromatin is the genetic material. If we talk about the cell division comprises of two phases, that is interphase and the M phase. So rest of the time, the cell, inside the cell, the genetic material is in the form of chromatin, which are filamentous or fiber-like structure. And for the less time duration, these chromatin form a structure like chromosome. We all know this is chromosome. We all heard of it. So this chromosome. So the conversion of chromatin into chromosome is done with the help of protein, which is called a histone protein. So let me write it down here. So histone protein helps in condensation of chromatin to chromosome. Is that clear to everyone? And after it become a chromosome, there's a special concentrated thing over here. Is called as centromere. Centromere is the structure which is present. A concentrated structure is present at chromosome, which is known as centromere, which has a huge significance in cell division. And the next thing is, I have explained nucleopore, I have explained nucleolus, nuclear membrane, and nucleoplasm, and histone protein also. So, there is also some topics remaining, that is shape of chromosomes. Actually, in our NCERT, 4 types of shapes are explained. Let me draw it first. This shape of chromosome, let me write it down. Shapes of chromosome, these are as follows. So this shape of chromosome, actually the shape of chromosome depends on the position of centromere on chromosome. So if you want to understand the shape of chromosome, you need to first focus on the centromere, where the centromere is present. So this is the basic chromosome you everybody have seen every time in NCERT. So this is the basic chromosome and this is the centromere. So this chromosomal structure is metacentric. Again, the next chromosomal structure in which, as you can see, these are shorter arms, we say arms, and this is long arms. This is known as submetacentric. Again, here we go. As you can see, centromere position has again changed, and as it goes closer to the terminal position, the name changes and the type of chromosome also changes. So this type of centromere position and this type of chromosome is known as acrocentric. The next one is like this. This is telocentric. So these 4 types of chromosome are explained in your NCERT as I have drawn on the board. So as you can see, from metacentric to telocentric, the position of centromere is getting upward, or we can say coming near to one end of a chromosome, like this, and this after that, and this. Here is another term explained in the NCERT, that is known as satellite DNA. So let me explain. So satellite DNA is a kind of DNA that is present on the terminal position of chromosome, like this. So this red spots are satellite DNA. Actually, satellite DNA is it is different from the rest of the DNA because when we just observe chromosome in electron microscope after dyeing with a basic dye, so the dye does not act on the satellite DNA. Actually, it is very difficult to see satellite DNA in electron microscope also. That is why, so a special type of special part of chromosome, which is known as satellite DNA, is present over here. That is known as satellite DNA. So it doesn't take color of dye. So that is why it is called as satellite DNA, because it is very difficult to see in electron microscope as well. So these 4 types of chromosomes are explained in your NCERT and in the topic nucleus. So I think I have covered each and every topic of today, and I think that the cell, the basic unit of life, comes to an end here. So if you have any doubt regarding this chapter, you can ask me anytime, anywhere. And first